Thank you very much. Okay, great, it's working. So I heard that I look quite similar to the previous speaker because we were mistaken the slides. So fittingly enough, I'm talking about phenotype similarity. So here are my conflicts of interest. So today I'm talking to you about inherited retinal diseases. So basically they are single gene disorders that affect the retina, and a mutation in any of 270 genes can cause this disease. Here are some typical patterns of degeneration that you can see on the left there. So here's the pointer, good. These are the corresponding genes underneath that are symptomatic of that pattern of deterioration. The reason why you get these different patterns is conceptually because different genes are expressed specific to different cell types, and depending on the cell type which is affected, this can lead to different manifestations of a disease and different patterns of progression. So I'm interested in identifying the genetic diagnosis of patients with these inherited retinal diseases. So for people who haven't heard about inherited retinal diseases, there's quite a few people in the UK who are affected by this, and actually they're a leading cause of blindness in working age population. Over 40% of the patients remain undiagnosed, and it's significant. There's a significant health economic burden for the UK, because this is a lifelong condition, and it can take a very long time for patients to get a diagnosis. The benefits of a timely diagnosis are significant for patients, for psychological support, prognosis, family planning. Also, if you have a genetic diagnosis, you might be able to detect a more serious life-threatening underlying condition. And also now we're getting more and more gene-targeted treatments. So more and more of these patients are eligible for treatment or can be recruited to a clinical trial. And it saves the health service, the health system a lot of money by knowing exactly what's wrong with these patients. Traditionally, single gene testing has been used, and then gene panel, and now more and more we're seeing whole genome sequencing be used to diagnose these patients. And actually, we heard about Genomics England previously from Jenny, and I think there's around 6% of the patients in the rare disease cohort are actually rare eye disease patients, so quite a lot of patients. We've also seen from Jenny that around the whole diagnostic rate is around 25% over the whole of Genomics England. For inherited retinal diseases, well, specifically for ophthalmic disorders, it's a bit higher, it's 50%, but it's still pretty good. We're working hard on trying to improve that diagnostic rate with various variant interpretation tools. We saw the nice work by Jenny. We're looking at RNA sequencing data. There's also a poster by Jamie Ellingford on looking at non-coding variants, I believe. So I'm going to be focalizing more on the PP4 criteria, which is in the ACMG, part of the ACMG criterion. So there's a lot of effort in trying to standardize the way we interpret genetic variants, and there's a lot of work going on behind the scenes with the ClinGen group, and that have divided into specialist working groups. There's a working group that is specialized on the ocular genes, for example. Now, one challenge we have with these criterion is that actually it's often the case that we get conflicting criteria, and that can lead to variants of unknown significance, as we've heard. I think around 10% of the variants in Genomics England are 10% of the reported variants are variants of unknown significance. So it's really important to be able to get better tools to give us a more precise answer to these precise interpretation for these criteria. So I'm going to focus on the PP4 one, which is the phenotype similarity. The importance of phenotypes is significant in Genomics England, and actually I think around 80% of all the variants, all the patients in Genomics England were solved with this automatic variant prioritization tool known as Exomizer. which takes as input the PED file, the family history, the VCF file, the variants, but also importantly the HPO terms, which are, I'll talk about in the next slide, which are basically a standardized description of the phenotype. And this is even more significant for the inherited retinal diseases, where actually Cipriani et al. Have seen that if you include HPO terms, you can get a diagnostic yield improvement of quite important, of around 40–50%. For those who are not familiar with the HPO, the Human Phenotype Ontology, briefly, it's a hierarchy of medical terms with more general, less specific terms at the top and more specific terms at the bottom of the hierarchy. There's around 1,139 terms, which are for abnormalities of the eye, of which 364 are for the abnormalities of the posterior segment, which means the retina. And of these, 37 are linked to, can be linked to imaging, linked back to imaging of the retina. So there's been a lot of work on curating this data, on curating these HPO terms, and that's work by Panos Guniotis, who's done a really nice job on that. However, the relationship between the HPO terms and the genes is not always very complete. Here what we're seeing is the HPO terms and their corresponding images. So we've got, you can see 4 examples here. For people who know a lot about inherited retinal diseases, they will notice that a few genes, for example, are not there, and some HPO terms don't have their associated genes with them. And also another challenge is that when the disease progresses, the HPO terms also change. So these challenges with HPO terms, basically, which are, well, incomplete information about gene associations, progressive conditions, and also I haven't even mentioned the time it takes to actually identify these HPO terms, has motivated our approach, which is to try and use AI directly on the retinal scans to identify the genes, rather than going through the HPO terms. So this is the idea behind Eye2Gene, which takes as input retinal scans and applies an AI artificial intelligence algorithm to prioritize which gene is the most likely to be causing the disease. To train Eye2Gene, we use the data sets at Moorfields Eye Hospital, which has a very large cohort of patients with inherited retinal disease who all have a genetic diagnosis and corresponding imaging. The reason why there's 3 bar plots here is because there's 3 different types of imaging. So we trained an AI, we trained AI algorithms to identify on the 3 different types of imaging. These are the total numbers of patients, including around 2,000. We've also published the cohort a few years ago, if you want to read the paper. And so we use this as a training data set, and we train these 3 different basically convolutional neural networks, or AI modules, if you want, in order to identify, from retinal scans, to identify the genes. And as you can see, it's combining, when you have the different types of imaging, it can combine the different types of imaging to give you a combined prediction. So it can use information from the 3 types of imaging if you have them available. So now, using that trained neural network, what we did is we looked at the penultimate layer of the network. So before it gives you the answer, the gene answer, and that's actually a very high-dimensional vector, 128-dimensional vector. So using that vector, we then reduced it down to 2 dimensions in order to plot it. So what you're seeing here on the right are these little gray points. Each of these represents a scan which has been fed into the Eye2Gene neural network, and you're seeing the 2-dimensional projection of that. So it's in a way you can think of it as looking at inside the brain of the AI and seeing what neurons or whatever get activated based on the input. And in red, we can see in red we have the genes, basically. So the scans from a specific gene which activates specific of these points. So using this, it's quite interesting to look at that, because what we can also notice is sometimes the points are either very clustered or they are quite separate. And the points being separate is underlying potentially different phenotypes for the same gene. So if you're interested in this sort of approaches, you can you can attend the talk by Hannah Klinkenhammer, I think on Monday, who's applying this type of technique to lump or split phenotypes. So we didn't go that far to lump and split the phenotypes, but we did use these points in order to apply a clustering algorithm, a bit like the phylogeny clustering you saw in the previous talk by Natalie. And using that, we obtain a dendrogram, and we're able to recapitulate a lot of the known phenotypes. So I've highlighted them there for you. So these are known phenotypes, so it's quite encouraging to see that Eye2Gene is identifying the phenotypes that are already known. Some of them are not known, but it's worth looking at this further. So this is for 70 genes, and this is for all the genes that we've got in our data set, which is 120. So using this approach, we wanted to evaluate it and see how does it compare with using HPO terms. So for that, a clinical fellow kindly reviewed notes and retinal scans of 130 inherited retinal disease patients with a confirmed gene diagnosis. And from that data set, a total of 21 distinct HPO terms were identified, 4.7 on average per patient. And each patient's HPO terms were used as input for the Exomiser 5 phenotype gene scoring algorithm. So as you've seen previously, the Exomiser algorithm uses both a VCF file, which is the genetic data, a PD file, which is the mode of inheritance, and the HPO terms. Here we're only looking at the HPO side of the Exomiser algorithm. So that's why it's called the Exomiser 5 phenotype gene scoring algorithm. And we also used we also obviously gave the retinal scans of those patients to Eye2Gene. This is the phenotype grid that we obtain. I'm sorry, it's a bit blurry, I think. But this is not all the patients here. This is like the 30 of the patients. And basically, the rows are patients, and the columns are phenotypes. And if there's a dark blue square, that means the phenotype is present in the patient. So we apply this, we use these HPO terms, these phenotypes, as input to the Exomiser 5 algorithm. And here what we've got on the right here now is the ranking of which gene was considered to, the ranking of the gene that was considered to be the top. So the actual gene obviously is known because we have the gene diagnosis. So for example, in the first case, it's USH2A is the gene, but the ranking by the Exomiser 5 algorithm is the second one. So it thinks it's the second. It's ranked as the second most likely. So we did the same thing with Eye2Gene, and here are the rankings compared, and we find that. Overall, 75% of the time, Eye2Gene ranks the correct gene the same or better than just using the HPO terms. So this motivates an approach which would combine both the traditional approach of variant interpretation using Exomiser and HPO terms with a combined approach of using Eye2Gene and retinal scans when available. So in conclusion, Eye2Gene can infer a causative gene directly from retinal scans without the need for HPO terms. These types of AI phenotype-driven approaches, which work directly from imaging, are becoming increasingly popular, and they can really, we can really make the most of the PP4 criterion using these approaches. There's many approaches. The most famous is probably Gestalt Matcher by Peter Kravitz, but there's also deep plasia, which is an approach that works on bone imaging. There's a poster that you can have a look at, and as I touched upon, it would be really interesting to use this AI approach to see if we can identify novel gene phenotype relationships and groupings. I was talking with Alfrede de Barra about this, and whether we could identify, for example, spliceopathy genes in that way. If you want to learn more about the project, you can go and check the project website and read the preprint. Thank you very much.
Thank you. Thank you very much, Dr. Ponticos. Unfortunately, we are over time, so those of you who would like to ask Dr. Ponticos a question, please come to us after the stage. I want to thank all of the speakers very much. I want to thank you and enjoy the rest of the conference. Thank you.